Welcome back to BioMindscape. So today we are going to know about the allelic frequency. That means a population consists of a particular allele in proportion. So for knowing that particular allele proportion, we use allelic frequency. In population genetics, it is very important. If a gene have a two alleles, for example, capital A is dominant and small a is recessive. These are the alleles and the genotype. There are three genotypes: capital A, capital A, capital A, small a, small a, small a. Where capital A is a dominant one, and small a is a recessive one, and capital A, small a is a heterozygote one. And each individual has two alleles in a diploid organism. Formula for allelic frequency is P. P is equal to frequency of capital A dominant allele, that is 2 in bracket capital A plus heterozygote, that is capital A small a, upon the total into 2 into the total number, and frequency of small a allele, that is Q, that comes out to be 2 in bracket the recessive alleles plus the heterozygote allele upon 2n, that is total individual. And also remember P plus Q is equal to 1. So this is the important formula. We will know everything by an example. So let's begin the example. Suppose the population is capital A is 40, capital A is 40, and small is 20. So we just have to add it. So it comes out to be 100. So total allele is 2 into 100, that is n. So is equal to 200. Now let's calculate the dominant alleles capital A. So capital A individual each has 2 into the dominant allele, that is 40 into 2, that is 80. Similarly for heterozygote we will multiply it by 1 allele because each has one allele. So 40 into 1, that is 40, and total allele, that is 80 plus 40, that is 120. So we just have to find the frequency of this total dominant allele. For that we will just divide the total number of dominant allele upon the total allele, that is 120 upon 200, that comes out to be 0.6. Now let's calculate the small a, that is the recessive allele. For that we have taken recessive individual gametophyte and each has a 2 allele. So 20 into 2, that comes out to be 40. If we have a heterozygous individual, then we will multiply 1 into the recessive allele, which comes out to be 40 into 1, that is 40. So the total number of recessive allele, small a allele, that is 40 plus 40, that is 80. So we have to find the recessive allele frequency. For that we will write Q is equal to 80 upon the total number of allele, that is 200, that comes out to be 0.4. So we get the final dominant and recessive allele. So we will add it like 0.6 plus 0.4, which comes out to be 1. Now if we have to find the genotypic frequency, we will just write it f of capital A frequency of the heterozygote, frequency of recessive, and then we will put in the formula P is equal to F in bracket the dominant number of alleles genotype capital A plus half into the frequency of the heterozygote genotype capital A small a, and for the similarly for the recessive we'll use the same formula, but instead of capital A we will use small a. So we can use a shortcut like P is equal to capital A genotype plus half into the heterozygote. Similarly for recessive we can use Q is equal to the recessive small a plus half into heterozygote when the values are already in the frequency form. So in a population if there are 200 individuals, the genotypic number are given here. We have to find the allelic frequency of the dominant and the recessive one. So total individual, that is total allele in a population. So the total individual that we get in a population of 200 individual, genotype number are capital A dominant 80, heterozygous 90, and the recessive genotype it shows 30. So we have to find the allelic frequency of the dominant and recessive. So total individual, that comes out to be 200, that is 80 plus 90 plus 30. So total allelic population, that is the 2n, that is 2 into 200, that is 400. So the number of capital, that is dominant alleles for the genotype capital A, it has 2 alleles. So 80 into 2, that is 160, and for recessive allele, that is 90 into 1, that because it has only one heterozygous allele, so that comes out to be 90. So we have to add it 160 plus 90, which comes out to be 250. So the frequency, that is P is equal to 250 upon 400, that comes out to be 0.625. Number of the recessive allele, similarly, that is 30 into 2, that is 60, and for the heterozygous it is 90 into 1, 90. So the total 60 plus 90 is 150. So we have to find out the frequency of the recessive allele, that's come out to be 150 upon 400, that is 0.375. So we got the allelic frequency of the dominant allele, that is 0.625, and the recessive one is 0.375. So we just have to add it and we get 1. You can directly calculate by another formula, that is P is equal to 2 into the dominant genotype plus the heterozygote upon the total number of allele into 2. And for similarly for the Q, that is the recessive one, the 2 into the recessive genotype plus heterozygote genotype upon 2 into capital N. Now let's frequency. That means the population has a particular genotype in a proportion or a fraction. So for knowing this, we need a genetic frequency. For example, if a population has 3 genotype like capital A, capital A, capital A, and small a, and the total individual, that is the n. So the genotypic frequency has a formula of f of in bracket the dominant capital A, that is equal to the number of capital A individual upon n, and for the heterozygote capital A small a is equal to the number of capital A small individual upon n, and for the recessive one frequency of in bracket small a is equal to number of small individual upon n. So the important rule is here is frequency of the dominant plus frequency of heterozygote plus the frequency of the recessive is equal to 1. So for suppose we have a population of dominant 50, heterozygous 30, and the recessive 20. So we just have to add it to get the total number, total population, that is 100, 50 plus 30 plus 20. So we have to calculate the genotypic frequency. For calculating the genotypic frequency, we will have the formula, that is individual like capital A upon the total population, that is 100, which comes out to be 0.5. Similarly for the heterozygote as well as the recessive one, this 30 upon 100, that is 0.3, and 20 upon 100, 0.2. So these are the three values that we get for dominant, recessive and the heterozygote. 0.5 plus 0.3 plus 0.2, that comes out to be 1. So now we can also find out the allelic frequency by this genotypic frequency. After finding out the genotypic frequency, we can find out the allelic frequency. How? We have to put a formula, that is P is equal to F in bracket capital A plus half of F of capital A small a, the heterozygote, like P is equal to 0.5 plus half into 0.3, that comes out to be 0.65. So this we got an allelic frequency of a dominant one, 0.5. Now by knowing this, we can also find out the recessive. Just by knowing the frequency of the dominant one, we can find out the recessive frequency, that is P plus Q is equal to 1 by putting in this formula 0.65 plus Q is equal to 1. So Q is equal to 1 minus 0.65. That's why the Q is equal to 3.35. So we can add P plus Q is equal to 1, so that comes out to be 1. So this is the correct answer. So 0.65 plus 0.35 is equal to 1. This is the allelic frequency for P and Q. For P it is 0.65 and for Q it is 0.35. So the allelic frequency is frequency of allele like P and Q, and genotypic frequency, frequency of a genotype like capital A, capital A, capital A, small a, and small a, small a. This is the genotype. And thanks for watching. Do like, share, and subscribe BioMindscape.